Depends on, again, that target product profile, where within that risk category do you need, different levels of these different items. But yes, that's a great way of looking at it.
Nick, let's speak today a little bit more about what you need to make a medical device. And we speak to a lot of people that are all very excited about making their inventions happen. And one of those things that they're often looking for is spaces where they can do that development. Now, I know that you have experience with setting up laboratories and clean rooms, and so I was going to ask you, if you think about that, what are typical spaces that you need for medical device development, and how does that relate to product development?
Yeah, very good. And again, this talks to a theme that we talk about here on the program. That's target product profile, what you actually need and when you need it. As it relates to biology, particularly what you and I do on a day-to-day basis, you really need to verify the function of your diagnostic or your assay, or even how the medical device engages with biology. Guess what, folks? We're biologic. And so just like a mechanic needs a garage where it's got a hoist and you can change oil and put the oil in the correct spot and not just dump it down the drain, the biomedical engineer needs a proper lab that's clean and safe, and you're not going to take the fluids and just dump it down the drain. And so essentially what you need is the proper containment. And there's different levels of containment depending on the risk category. It's very much like engineering that you want to work within. So we have these different, what we call biosafety level groups, and that's the lowest or least risky being BSL-1, and things like yogurt, E. coli, things of that nature you can handle in that environment. It works its way up. BSL-2 is a little bit more complicated, things that will get you sick if you get a high enough dose and even potentially kill you. And so you want to make sure you've got different levels of containment. You work in biocontainment hoods, you have proper PPE and proper procedures. Then you get BSL-3, which is even more risky, right? So things like tuberculosis, if you're going to be handling that, it's very, very infectious, low infectious disease. You have the same levels that you had at level 1 and level 2, but you add an extra layer and you shower in, shower out, everything's in containment. And then you get to the granddaddy of them all, level 4, and that's the positive pressure spacesuits. And that's reserved for things like Ebola and filovirus.
That's probably not so fun to work with.
Not so fun to work with, and frankly, there's only one site in Canada that actually does that in Winnipeg. So depending on the target product profile, the risk factor that you're working with, you want to work within those different containment levels. Here we use CL2 or biosafety level 2 for our facilities. And you want to be able to do the right amount of testing with the right amount of risk and not overburden. Like, you don't need to be in a spacesuit to do a lot of the work that we're doing. The other thing that we do is when you're doing the assembly work, you need to do that in a clean space. And just like with the biosafety containment levels, that ranges from ISO 8, which is basically room air with a certain particle count, all the way up or down, depending on how you go with particles, to ISO 1. And that's, again, spacesuits, ultra clean air, and really, depending on what you're doing, you don't really need that. So having the right fit for the right development.
Yeah. So when you're describing a BSL-2 laboratory space, you use that mostly for testing. So you can think of that as a testing space or a space that's used during development for testing.
Correct.
And a clean room would really be used for the production of medical device parts, or the whole medical device assembly process.
Yeah, absolutely. And again, it depends on, again, that target product profile, where within that risk category do you need different levels of these different items. But yes, that's a great way of looking at it. And what we like to do here, we're kind of in this, what I call kind of the sweet spot. We're doing kind of preclinical or early clinical phase work, and so we're not all the way scaling up to full-on production of millions of, say, lateral flow chips or whatever. We're making smallish 1,000 units for early clinical trial, early verification validation work to make sure that the device is fitting within this target product profile. That's critical for success.
Okay, well, thanks. That's very informative.